My name is Kylie Stazewski. Today is March 26th of 2025, and this is my presentation on Chapter 55, Hemostasis and Thrombosis. This presentation will describe the fundamental aspects of platelet biology described in Chapter 55, Hemostasis and Thrombosis, as well as the basic characteristics of the proteins of the blood coagulation system and fibrinolysis. In addition, the biomedical relevance of these topics will be discussed. So, let's first begin by talking about hemorrhagic and thrombotic states. Hemorrhagic and thrombotic states can cause life-threatening emergencies if they're not treated promptly. A rational and clear understanding of these conditions is crucial to modern medicine. For example, internal bleeding or external trauma are examples of hemorrhagic states. In comparison, conditions such as pulmonary embolism, stroke, and heart attack are all examples of thrombotic emergencies. Thrombosis of the cerebral and coronary arteries are the major causes of death in today's society. So what is hemostasis and what is thrombosis? Hemostasis is defined as the cessation of bleeding from a cut or severed vessel, whereas thrombosis is the damage or removal of endothelium lining blood vessels. Both of these processes involve blood vessels, platelet aggregation, and plasma proteins that cause the formation or dissolution of platelet aggregates and fibrin. Hemostasis and thrombosis share three phases. In hemostasis, there is initial vasoconstriction of the injured vessel, causing diminished blood flow, which is distal, or away, to the injury. The first phase is platelet aggregation, in which platelets bind to collagen at the site of the injury and release ADP, which attracts other platelets to the area. Thrombin, which is formed during coagulation at the same site, causes additional platelet activation. When activated, the platelets change shape and in the presence of fibrinogen or the von Willebrand factor, aggregate to form the hemostatic plug during hemostasis or thrombus during thrombosis. Now that we have discussed the basics of thrombosis, we can begin to discuss the types of thrombi. There are three types of thrombi: white, red, and fibrin. The white thrombus is mainly composed of platelets and fibrin and is generally poor in red blood cells, also known as erythrocytes, while white thrombi tend to form at the site of an injury, of deformed or damaged vessel walls, and are common in areas where blood flow is rapid, such as arteries. In contrast, the red thrombi primarily consist of red blood cells and fibrin and often form in areas of static blood flow, such as blood tubes in a lab or pulling injury. Red thrombi are the typical blood clots that form in lab tubes. They occur with or without injury. Finally, fibrin thrombi are composed solely of thrombin and occur in small blood vessels or capillaries. Now we are going to move on to platelet aggregation. There are three steps to platelet aggregation, which include adhesion, where platelets are exposed to collagen blood vessels, exocytosis of storage granules, and aggregation. First, platelets use receptors to adhere to collagen via specific receptors called glycoprotein complexes on their surfaces. The binding of those complexes to collagen is mediated by something called the von Willebrand factor, which is a large multimeric protein that plays a role in blood clotting. Collagen-bound platelets change their form and spread out on the subendothelium. Next, they release the contents of their storage granules. This process is stimulated by the enzyme thrombin. ADP is then released. The released granules, which activate platelets with specific G protein-coupled receptors, resulting in the aggregation of additional platelets. Endothelial cells located in the walls of blood vessels are vital to the regulation of hemostasis and thrombosis. Endothelial cells are responsible for the synthesis of a lipid molecule called prostanoid prostacylin, which functions as a vasodilator. In addition, endothelial cells express protidoglycans such as heparin sulfate, an anticoagulant, and they also release plasminogen activators which work to dissolve thrombi. Antiplatelet drugs are used to inhibit platelet responses within the body. The most commonly used antiplatelet drug is aspirin. Drugs such as aspirin inhibit the enzyme coaxylase, also known as COX-1, which is involved in the formation of TXA2, which is a vasoconstrictor and a common aggregator of platelets. Antiplatelet drugs are typically used for the management of acute coronary syndromes such as myocardial infarction, carotid artery stenosis, angina, and even acute stroke syndromes. Other commonly used antiplatelet drugs include clopidogrel, prasugrel, and ticagrelor. There are two pathways that lead to fibrin clot formation, the intrinsic pathway and the extrinsic pathway. It is important to note that these pathways are not independent of one another. The intrinsic pathway is activated by negatively charged surfaces that enter the body. For example, a shard of glass that would cause damage to vessels would trigger the intrinsic pathway. The extrinsic pathway is triggered by tissue damage. More specifically, this pathway is triggered by a protein known as tissue factor, abbreviated as TF, found on the surface of damaged cells and is exposed to blood. Thrombin must carefully be regulated within the body, and this is achieved by one of two ways. The first is by feedback mechanisms, the second is by circulating inhibitors. Feedback mechanisms rely on the delicate balance of activation and inhibition. Thrombin circulates as its inactive precursor prothrombin. Prothrombin is activated by multiple enzymatic reactions and thus converted to thrombin. In contrast, circulating inhibitors such as antithrombin are regulated in the presence of heparins. Heparins regulate the amount of thrombin in circulation by binding to cationic sites of antithrombin and thus promoting the binding of antithrombin to thrombin. Heparin and warfarin are common antithrombotic drugs and are commonly used for the treatment and prevention of excessive clotting or clotting disease. Common conditions requiring treatment with antithrombotics are deep vein thrombosis, stroke, or pulmonary embolism. Heparin is also commonly used as an anticoagulant additive in blood tubes to prevent samples from clotting. Now that we have discussed how blood clotting works, we can discuss the diseases of the coagulation system. Hemophilia is a disorder of the clotting system, and thus individuals with hemophilia typically experience excessive bleeding from small scrapes, bruising, dizziness, and common nosebleeds. The term hemophilia can literally be translated from Latin to English as blood love, meaning that individuals with hemophilia love to bleed. Hemophilia A and B are genetically inherited deficiencies linked to the X chromosome. The most common type of hemophilia is type A, which is caused by a deficiency in clotting factor 8, while type B is extremely rare and caused by a deficiency in factor 6. The most common hereditary bleeding disorder is von Willebrand disease, which occurs in up to 1% of the general population. Von Willebrand disease results in a defect or deficiency of the von Willebrand factor, which is a large multimeric glycoprotein secreted by endothelial cells and platelets. The glycoprotein is responsible for stabilizing blood clotting factor 8, much like hemophilia. Von Willebrand disease results in excessive bleeding, bruising, and can prolong recovery from minor injuries. Now we can talk about diagnostics. So there are various laboratory diagnostics used to measure the factors contributing to hemostasis and thrombosis. The skin bleeding test is an overall test of platelet and vessel wall function, while the closure test is measured by using the platelet function analyzer PFA, which is an in vitro test of platelet-related hemostasis. The platelet aggregation test is used to measure the response to specific aggregating agents. A PTT is a measure of an intrinsic pathway and PT of the extrinsic pathway, with the PTT being used to monitor the effectiveness of heparin and warfarin. Now we've come to our conclusion. This presentation has summarized the topics described in Chapter 55, Hemostasis and Thrombosis, including platelet biology, protein biology, coagulation factors, blood vessels, and the common diseases and syndromes of the pharmaceuticals used to treat and manage the diseases of the blood coagulation system. In addition, the biomedical relevance of these topics were discussed, and the various ways that these disorders are tested for. That brings us to our multiple choice questions. Our first question here is, what pathway is activated as a result of exposure to negatively charged particles? The correct answer here would be option A, intrinsic. So, as we remember from slide 10, the intrinsic pathway is activated by negatively charged surfaces that enter the body. Question 2. What type of thrombus is primarily found in small veins and capillaries? The correct answer here is option C, fibrin thrombus. This information was found on slide 6. Now we have question 3. What is the most commonly used antiplatelet drug? And the answer is aspirin. Now we have question 4. In which phase of platelet aggregation does exocytosis occur? If we remember here, the answer is option A, the release phase in which all of the granules exhibit exocytosis. Now we have question 5. Which of the following laboratory diagnostics measures the effectiveness of warfarin and heparin? The answer here is C, APTT. All of my references were taken from our textbook, especially chapter 55, Hemostasis and Thrombosis. Thank you.